My name is Colin Schuman. I'm in Biology 200, and we're going to be talking about proteins. So, what exactly is a protein? Protein Proteins are composed of long chains of amino acids forming polypeptides. An amino acid is a molecule that is made up of a carboxyl group, which is shown right here, in addition to a bond between oxygen and hydrogen over here, a central carbon, which is always connected to a hydrogen atom, shown right here, and an amino group, which is a nitrogen bonded to two hydrogens. So you can see that right here, and you can see that the hydrogen has been removed right here, and an R group, which varies in amino acids and ultimately determines what an amino acid is. So these long chains of amino acids are called polymers, bonded by peptide bonds. This is an example of primary structure, which is the amino acid sequence of a single polypeptide chain. The amino acids in the middle between the N terminus, which is on this side, and the C terminus, this side, determine the protein. The N terminus is at one end of a peptide chain, which is composed of a nitrogen atom and three hydrogen atoms. This connects to a chain of amino acids right here. On the other side is the C terminus, which is a carbon double bonded to an oxygen, and a negatively charged oxygen on the side. Primary structure is the strongest out of all the structures due to its covalent bonds. Next, we have secondary structure, which is the localized area of repeating structure. This includes the alpha helix, shown right here, and the B-pleated sheet, which is shown right here. What occurs is that the same peptide chain bonds to itself, so you can see the same chain is bonding to itself through hydrogen bonds between the carboxyl groups of the amino acid, where the carbon is double bonded to the oxygen, is where the bonds will end up. So you can't see that in this model, but the carbon is double bonded to the oxygen. Although hydrogen bonds are weak on their own, lots of hydrogen bonds along the peptide chain allow for the strong unified bond, shown right here and right here. The alpha helix is shown right here, the B-pleated sheet is shown right here. And as you can see, the hydrogen bonds form right here, and the hydrogen bonds form right here. Next, we move on to the final 3D shape of a single polypeptide chain, the tertiary structure. There are multiple types of bonds, such as ionic, hydrogen, and hydrophobic bonds, but they are relatively weak. Hydrogen bonds will form between the side chains, so right in here. Ionic bonds will form between charged amino acids and charged R groups, so within the polymer. However, there can be covalent bonds between two sulfate atoms that happen to join together between R groups. This disulfide bond can be very strong. Lastly, we have the quaternary structure. This is the final 3D shape of two or more polypeptides. This includes two or more of the tertiary structures, which can be shown here, 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 and here, coming together to an overall structure. So all of them are coming together into one. They will have the same overall weak bonds as the tertiary structure. Quaternary structure is the overall product where the protein can become functional based on the shape of the structure. So here we have four types of long tertiary structures coming into one. So 1, 2, 3, 4. We can see the individual tertiary structures. It takes a chaperone protein to help fold the proteins into thermodynamically stable 3D shapes. But the end products of a quaternary structure is a bunch of different polypeptide chains joining together to form its final shape, which ultimately determine a protein structure. Thank you for listening.